Did you know that a cell surface membrane drawn with a single line is actually a phospholipid bilayer? In fact, all of the membrane-bound organelles are phospholipid bilayer. Organelles such as mitochondria and nucleus, on the other hand, have 4 layers of phospholipid as a result of consisting of 2 layers of membrane, an outer and inner membrane, each of which is a phospholipid bilayer of its own. Organelles such as the ER and the Golgi body, on the other hand, consist of a single phospholipid bilayer with high percentage of unsaturated fatty acid tails, so that only then it is more fluid for vesicle budding and fusion. Phospholipid bilayer of the lysosome, on the other hand, consists of dense amount of glycosylated membrane proteins so as to protect the bilayer from being digested by the luminal hydrolases. In all cases, the phospholipid molecules that make up each of the phospholipid bilayer consist of a glycerol backbone that has been linked to a phosphate group and 2 fatty acid chains that can be saturated or unsaturated. It is the phosphate group that is being represented by the polar hydrophilic head, while the fatty acid chains represented by the 2 tails of a phospholipid molecule. A liposome with such configuration of phospholipid molecules can be prepared artificially by subjecting a solution of phospholipid to sound waves, a process known by sonification. They possess a lipid bilayer and an internal aqueous environment which can be loaded with a drug for the targeted delivery of drugs and to minimize unwanted effects in non-target tissues. It is also used for the delivery of nucleic acids in gene therapy trials. Components like antibodies can be incorporated into the liposomal membrane so that they are targeted to specific tissues. After emulsification, phospholipid bilayers become micelles, which consist of a single layer of phospholipids with hydrophobic tails enclosed within. Lipids are absorbed in intestine in the form of micelles.